Hello students. So today we'll discuss about the part second of the transpiration. That is chapter 4. In the part first we will discuss about the transpiration, its definition, introduction, and its significance, and its three types. Okay, and the structure of stomata also. So today we will discuss about the mechanism of opening and closing of stomata. So basically the process of opening and closing of stomata is known as stomatal movement. So in the structure you can see the inner wall is thick and non-elastic, while the outer wall of the stomata is thin and elastic one. So it contains a guard cell. So these guard cells itself contain the starch with chloroplast, while surrounding epidermal cells do not contain starch with chloroplast. During the daytime, so when there is a light, starch is converted into sugar in the presence of inorganic phosphate. So during this process, an enzyme known as phosphorylase act as a catalyst in this. So glucose being soluble in water increases the osmotic pressure of the guard cells. As a result, guard cells takes water from the neighboring cells and diffusion pressure is formed in guard cells, which becomes turgid one. So the outer wall of the guard cell being thin and elastic gets stretched, or we can say pulled outward, while the inner wall is non-elastic and thick, which becomes concave in shape. As a result, these two guard cells move apart from each other and stoma gets open during the daytime. When the stoma are open, water vapor from inner cells, which we say intercellular cells, is lost to the outer atmosphere. Okay, it happens in the daytimes, whereas in the during the night times or the darkness, we can say sugar present in the guard cells is converted into a starch in the presence of enzyme phosphorylase. In the darkness it is formed. So the starch being insoluble and therefore osmotic pressure of the guard cell decreases. So as a result, water diffuses from the guard cells to the neighboring cells and turgor pressure decreases. So due to this decrease in the pressure of the guard cell, stoma is partially or completely closed during the night time. So when stomata are closed, the water in vapor form from the intercellular or the inner cellular space is not lost through stomata to the outer atmosphere. So the conversion of sugar to starch and starch to sugar depends on the pH and the temperature of guard cells. So at the daytime, pH of the guard cells remains 7, which is a neutral one or alkaline, and at the night time pH remains 5, that is a acidic one. So there are various theories have been developed by the various scientists. According to the first, we'll discuss about the Steward Hypothesis. So in Steward 1964 proposed the hypothesis to explain the mechanism of opening and closing of stomata. So according to this Steward hypothesis theory, so glucose 1 phosphate is osmotically inactive unless it is converted to glucose. So no change in osmotic relation can occur. So he showed that metabolic energy, that is ATP, is necessary for close of stomata. So during the daytime or in the light, we can say the starch becomes soluble and converted into soluble sugar with the help of phosphorylase enzyme. So this osmotic pressure and DPD, that is the diffusion pressure defeat of guard cells, absorb water from the outer cells and becomes turgid. So due to this turgidity, outer thin walls expands while inner thick wall stretch, and thus opening of stomata takes place. In the reaction we can find out the opening of the stomata. Okay. While in the night time, we can say the darkness, according to the Steward, the soluble sugar converted into starch. So it lowers the osmotic pressure and the diffusion pressure defeat, which in result releases of water from guard cells into outer neighboring cells. So thus thick walls come very close to each other and result in closing of stomata. So the turgor pressure plays key role in this opening and closing of stomata. So according to the Steward's belief that glucose 1 phosphate further should be converted to glucose to increase the osmotic pressure. So in this again ATP is essential for this conversion according to the Steward hypothesis. So thank you very much.